Assalamu alaikum. Honorable head, honorable external, honorable supervisor, and my dear audience. Assalamu alaikum. Thanks for giving me a floor to explain my research work. So, my research topic is about investigation of the status of heavy metal content and associated health risks in edible chicken parts supplied in Khulna city market. So, at first, the background of the study. At first, we have to know what is heavy metal. The metal and metalloids having atomic weight between 63 to 200 gram and whose density is more than 5 gram per centimeter cube are defined as heavy metals such as lead, chromium, cadmium, copper, nickel, etc. Then heavy metal has interesting and important characteristics, which is bioaccumulation and biomagnification nature. Human exposure to such metals have lead to severe toxicological implications. Then Bangladesh Council for Scientific and Industrial Research stated that poultry meat in local markets, in local Bangladeshi markets, are 2.5 milligram per liter chromium. Which is compared to greater than the WHO and FAO standard, which is 0.4 milligram per liter in chicken meat. Not only that, there are also some 30 type of harmful chemicals and also sodium chloride, which are used for tanning, are also found in chicken meat. Then health effect of heavy metal poisoning in chicken meats are reduced intellectual quality, anemia, neurological damages, nerves disorders, and also can be found cancer risks and many more. Thus, there is a possibility of human health risk by consumption of this type of chicken meats. Okay, therefore, this study has been conducted to assess the quality and quantity of heavy metals in chicken and also their harmful effects in both ecology and both in human health. So, now aim and objective of my study. At first, I tried to estimate the level of heavy metals accumulated by measure 4 types of chicken, which are broiler, Sonali, cock, layer, which are available in the major markets of Khulna City Corporation. Secondly, I tried to determine the heavy metal concentration in the feed, which feed they were actually fed, the chicken were actually fed. Then thirdly, I tried to compare the level of concentration of heavy metal accumulated by these body parts of different type of chicken, such as flesh, bone, liver, gizzard, and also in feeds with the WHO and FAO permissible limits. Fourthly, I tried to evaluate the health risk of human and assessing their potential ecological risk. Now, materials and method, the study area. Mainly 8 different farming areas were selected for collecting the chicken samples. These areas are Digholia, Daulatpur, Beara, Srifaltola, Gollamari, Botiaghata, Koya, and Pabla. My study period is although these areas are situated outside of Khulna city, but chicken rare in this area are supplied mainly in Khulna city market. So these areas were selected. My study period was March to May 2023. Now sample collection. 4 types of most common chicken found in Khulna city markets were collected as my sample. These are broiler, cock, Sonali, and layer. A total of 13 chicken were collected for lab analysis. These are 4 broiler, 3 cock, 3 Sonali, and 3 layer chicken. Not only that, their body part also analyzed, such as flesh, bone, liver, and gizzard of every chicken's body part was analyzed. Not only that, the feed of each chicken which they were fed also collected and analyzed. Also, broiler liver and intestine parts were analyzed because boiler litter, we know boiler litter and intestine samples are mostly used as fertilizer in agricultural land rather than the other chickens. So a total of in all a total of 60 samples were collected for this study. Now measure analysis and calculation. This is the main focus of my study, is that to determine the heavy metal concentration of all the 60 samples. From the literature review, I found that most critical heavy metals for this reason in chicken meat and bone and feet also. These are lead, chromium, cadmium, copper and nickel. So by finding this heavy metal concentration, actually two type of risk was assessed. First is ecological risk assessment and the other is health risk assessment. In ecological risk assessment, as I mentioned earlier, only boiler litter and intestine samples were collected for this research, because these are applied mainly rather than other cock or sonali or layer chicken. In agricultural land, when they are dumped, then soil was contaminated. Soil is being contaminated. So soil contamination factor can be found by dividing the concentration of heavy metals by their background values. Then health risk assessment. Two type of mainly health risk was assessed. First one is non-carcinogenic health risk. This type of health risk are not occurring cancer cells, but carcinogenic health risk mainly denotes the probability of developing cancer cells. Under non-carcinogenic health risk, four type of values were identified. First one is bioaccumulation factor. The bioaccumulation factor was derived by concentration of chicken part divided by concentration of their feed. Then estimated daily intake. Estimated daily intake was defined by multiplying concentration of heavy metal into their ingestion rate divided by their average body weight. In Bangladesh, average body weight was considered at 49.5 kg per body from the literature review. Then target hazard quotient. Target hazard quotient is defined as the division of edible estimated daily intake divided by reference dose. Here reference dose is the minimum acceptable oral dose or toxic substance below which no adverse non-carcinogenic risk will happen to human health. Then hazard index. Hazard index is simply adding a summation of the all total hazard quotient of all type of heavy metals in a chicken sample. Then carcinogenic health risk, as I mentioned earlier, it is the probability of developing cancer cells. So it can be expressed as target cancer risk. So target cancer risk can be determined as estimated daily intake multiplying with cleavage and polyadenylation specificity factor. Here I mentioned major findings of my thesis. At first, heavy metal concentration of in flesh part of different type of chicken. Here heavy metal concentration on different flesh part of different type of chickens were expressed. So with the permissible limits, the last red line denotes the safe limit by WHO and FAO. So it is clearly visible that all type of samples have excess values, excessive heavy metal concentration from the permissible limits showed in a red bar. So this is the heavy metal concentration of bone parts of different type of chicken. So here we can see the same results as they exceeds in many times than the permissible limits by WHO and FAO. Then the heavy metal concentration in liver parts. Here we can see also the same result, but somehow in luckily cadmium, for the cadmium, the beyond the threshold level, all type of heavy metals are beyond the threshold level, but others remaining the same results. Now heavy metal concentration in gizzard parts of different. Here also the same result, but here we can see lead and chromium are showing much more greater than other heavy metals. Now heavy metal concentration in liver, intestine and liver and intestine part. Here cadmium is lower than the permissible limits, but copper and chromium are excessively high. Here also in chicken feed, chromium, copper, lead, nickel, all heavy metals are greater than the permissible limit except cadmium. It shows the level beyond threshold. Now ecological risk assessment. Here I try to show mean contamination factor of heavy metal in boiler, litter and intestine samples. These values are expressed in that graph. That shows much more clear. The heavy metal beyond 0.10 to 0.25 in lead shows the slight contamination. The chromium and cadmium shows the slight pollution. Copper showed moderate pollution and nickel showed moderate contamination. This the red bar denotes their difference category. Okay. Severe to very severe slide. Okay. Here we can see the copper is excessively high in every broiler, litter or intestine part. That cause moderately pollution. Okay. Now non-carcinogenic health risk. So at first bioaccumulation factor. All the samples, chicken samples, the 13 chickens that are collected were quite much mature. Broiler was more than 40 days. Sonali or cock sample age was more than 60 days. Layer was more than 6 months. So bioaccumulation of their feeds has a great impact on them. This table indicates their bioaccumulation factors. In the right side, the remark is which bioaccumulation factor is greater than 1 are denoted with green bar. The green bar indicates that the indication of increased level beyond threshold. That is, their bioaccumulation factor is much more dangerous than other. Okay. Now estimated daily intake. The red bar shows their pollution level or threshold beyond 1. They are the minimum contamination with them. But 1 to 5 level is estimated daily intake divided by. Here is estimated daily intake divided by their digestion factor divided by digestion factor is the. Here is the main equation. So in this equation the result beyond 1 is expressed the minimum contamination, then 1 to 5 express the low health risk, and 5 to 10 indicates the moderate health risk, and higher than 10 indicates the severe health risk. Okay. Then target hazard quotient. According to United States Environmental Protection Agency, the acceptable guidelines for target hazard quotient has been reported to be 1 or less than 1. So here the target hazard quotient which are more than 1 are marked with red star. Here we can see the chromium, all chromium values and one copper value, which is cock liver. They shows the red star marks. They are above 1. So they indicate that there is a possibility of developing potential health risk. Now hazard index. Hazard index is mainly the simply summing of total target hazard quotient. Then if hazard index is less than 1 or 1, then a safe state for human body, whereas hazard index is more than 1, then non-carcinogenic health risk can be occurred. So Koch samples showed severe non-carcinogenic health risk, we can see, whereas the other shows relatively low. Because the layer is showing 3, Koch is whereas Koch is showing 6, then Sonali is showing 2.86 and boiler is 3.12. So here you can see decreasing level or order of hazard index is Koch more greater than boiler, then layer, and then Sonali. So here carcinogenic health risk assessment as a target cancer risk. Among these study levels, only heavy metal, lead was found the carcinogenic. Then, according to New York State Department of Health, stated that when target cancer risk is tends to 10 to inverse 3 to 10 inverse 5, then it can be called as a in a target cancer risk. So all the boilers, Sonali, Koch, and layer samples were were in target cancer risk with lead heavy metal. Okay, now conclusion. So observed concentration of heavy metal in all edible part of boiler, Sonali, Koch, and layer, who is all metals are higher than the Food and Agricultural Organization and WHO and also European Commission permissible limits. Number of values among them are exceeds safe limits, and these indicate that moderate to high health risk or probability to develop potential health risk. So heavy metal in exceed permissible limit in chicken layers, they are dumped as a fertilizer in village cultivation soil, or can be used as fish feed. Somehow they can enter in the human food chain. So bioaccumulation will be occurred. So use of heavy metals in chicken feed can be the first and foremost cause of heavy metal overload in chicken. In observed all fish in locally produced and also observed that 100% precaution was not taken during feed preparation, packaging, and marketing. And to lowering the contamination and controlling the pollution level of chicken edible parts, the whole processes like breeding, feed production, or growing up the chicken, all the circles should be monitored and under controlled by responsible authority. So result of this study can be used as an important database for further studies in this field or in this region. Thank you.